Welcome, everybody. Thank you so much to everybody for coming along to the International CML Foundation Forum. Can you hear clearly at the back? Yeah. So we thought this year we'd change the focus a little bit in terms of just trying to get a vision of where we should be trying to move over the next 5 to 10 years to get us closer to cure in chronic myeloid leukemia. I don't think any of us expect we're going to achieve cure in every patient in every circumstance, but it's something we need to be aspiring towards. So we thought we would put a few speakers to have a chance to put their own vision about that and then have a discussion. Really keen for all of you to be involved in that. Give us your own ideas about where we should be heading. So firstly, you'll realise that I'm not Nicola. Unfortunately, Nicola couldn't make it to the meeting, which is very sad. She won't be making it to the ASH meeting at all. And there's meant to be a whole bunch of brochures here as well, which don't seem to have arrived. So things haven't worked smoothly, but that's okay. We soldier on. So this is the format for the meeting. We have, following me, we have 3 really great speakers that are going to give us their perspectives. Mike Deininger, you all know very well. A provocative talk about treatment-free or free treatment. Ted Braun, who works closely with Brian Drucker, is giving us a talk about persistence in CML. And of course, Jorge, if and when he arrives, will be talking about resistance in CML. He did tell me he'd be a few minutes late, so I'm not at all worrying yet. And then Delphine Ray, very kindly, is going to join us on the stage for a discussion, as well as Lisa Machado. Is Lisa here? Oh, wonderful. Thank you. So Lisa is Executive Director of the Canadian CML Advocates. Is that accurate? So we clearly wanted to get a patient perspective on this question as well. So it'd be great to have Lisa and Delphine joining us for that part of the discussion. These are my disclosures as of today. So, I mean, I think we all recognise that there are still huge challenges for the patients and the clinicians in the low, middle-income countries. They're obvious. I don't need to go through them in detail. Availability of drugs, particularly second-generation drugs up front, third-line options, even second-line options in many cases, effective salvage options, monitoring. Obviously, we want accurate, standardised, sensitive, but we also want affordable. And as one person has suggested, point-of-care testing would be a huge step forward, particularly if there's significant delays in getting PCR results. Adequate health resources, and of course we shouldn't forget the fact that treatment-free remission should also be an aim and a pathway that we could work towards in countries that don't have the most advanced availability of drugs and PCR testing. So that's going to be a challenge for us. Today, I guess we're focusing more on what are the global challenges, and I've just plucked out 5 that I think are, from my point of view, going to bring us closer to cure in more patients, or at least to treatment-free remission. And I'll show you data that suggests that achieving uniformly rapid leukemia depletion really does have a substantial impact on the chances of achieving treatment-free remission. Secondly, I think there's a lot of evidence now that the immune system has a major role to play in the control of CML, and we need to understand how we can therapeutically enhance that. That's not going to be something we talk about in great detail today, but we can certainly deal with that in the discussion. I think we all believe that treatment-free remission needs to be achieved in more and more patients, but I think patients will tell us that they want to achieve it early, not waiting 5, 6, 7, 10 years before that can be achieved. Also, when patients stop drug, they don't want to face the possibility of treatment-free remission failure. Even though they go back on drug and they can get back to a remission, it's a traumatic process, and if we can minimise that, that would be a major step forward, getting more patients willing to take that step to stopping their drug. And we must not forget, and this will be dealt with in some detail by Mike Deininger, more effective, and also by Jorge Cortez, more effective, safer options for resistance and progression. So today, if you're just really oversimplifying where we stand, around about 20% of our patients have resistance or progression, and we really fall very short for those patients. Another 20% achieve good response to a level where they can maintain long-term, in many cases, major molecular response, but they can't become eligible for treatment-free remission with their current therapy, and they're sort of in a limbo of needing their drug long-term. Then we have the 60% who are eligible for treatment-free remission, and at the moment, around about half of them can achieve treatment-free remission and half cannot. So that's, I guess we're doing very well for 30%, but we need a lot of new solutions for the other 70%. So this is data that Naren Shanmuganathan, who's in the audience, is a colleague of mine, has put together from the Adelaide experience. And this is looking at all our patients, over 400 patients, looking at the cumulative incidence of actually being eligible for treatment-free remission. That is what we call having a deep response, MR 4.5, for at least 2 years. And you can see that that climbs steadily, and it's still climbing actually after 14 years. We still have more patients being recruited into this TFR-eligible cohort, which I think is encouraging. And if we look now at the patients who've attempted treatment-free remission, we see that the vast majority of patients in Adelaide who are eligible have actually gone on to attempt treatment-free remission. And as you might expect, about half have been successful. So you can see now that at 14 years, we have around about 40% of our patients who are achieving treatment-free remission. So I think this shows the good and the bad of the story. We're still getting more and more patients to treatment-free remission, but we're talking about 14 years before we can get them to the point of 40%. Now, of course, those patients who are at that 14-year period started their drug 14 years ago, so they're almost all on imatinib therapy. So I think we're going to see a lot more rapid achievement of treatment-free remission when we look at patients starting second-generation drugs. And we're going to have a look at that next to see whether I expect that this curve will move back about 5 years using the more potent drugs, but we're yet to see that. So this is the challenge that we have. Clearly, treatment-free remission is the optimal outcome, but it's only achieved today in the minority, as we've already seen. There are high failure rates, still around 50% in most studies, and of course, frustratingly requires many years of therapy. So the first two requirements are increasing eligibility and increasing the success once we stop. I think there's strong evidence that the second-generation drugs do increase the number of patients who are eligible, and they also have a similar rate of success. Therefore, overall, you're going to see more patients with treatment-free remission. Then the next challenges are to decrease the duration of therapy necessary to get patients to stop. And that's something that we're going to need to use new approaches to achieve that. And of course, as we've already seen, we need rational selection of who can stop and when. And finally, we need, obviously, to have safe and appropriate management. And something that there's increasingly looking at is the fact that the monitoring is actually quite burdensome for patients. Having monthly monitoring for 6 months and then 2 monthly for 6 months and then 3 monthly thereafter is probably excessive, and we're looking now at ways that we can reduce that to a more manageable monitoring regimen. So what are the best predictors of treatment-free remission? We know the strongest still is the duration of TKI, or the duration of deep molecular response. That's been shown in many studies, including the Euroski study, probably the biggest and the most important, but also other studies which I'll show in a moment. I want to give some of the data on halving time because I think it's very important to understand how that impacts on the chances of treatment-free remission. The transcript type does have an impact as well, and there's mounting evidence that the immune parameters are very important, but they're complex and they're not today enough understanding about that to be able to use that to decide who can stop and who can't stop. So this is the most recent data that we've seen that's come from the MD Anderson, showing that the duration of deep response, MR4, is very, very important to determine the success of treatment-free remission. So that patients that had more than 5 years of MR4 had a success rate of over 80%. Patients with shorter duration of MR4 significantly less. So that's an argument for being patient, but I think that's not an argument that our patients want to hear. They really want to find ways that we can bring our patients to a successful TFR in a much shorter time than 5+ years. So I wanted to spend a couple of minutes explaining this idea of halving time. So this is an example of two patients. One of them goes from 100% down to 10%, so it's a 1 log fall over 3 months, and one of them falls from 100% down to 0.1%. And so that's a 3 log fall. And if you calculate the halving time of those two patients, it's 27 days for the slow decline and 9 days for the rapid decline. And that represents a fairly typical range of most patients who start therapy, a halving time between around 9 days and 27 days. But of course, this is assuming that the patient starts at 100%. And many patients start at different levels. And if you're using appropriate PCR techniques, you can measure the actual level that they start at. And so these 3 patients who all end up at 1% at 3 months, because they had different starting values, 300%, 100% and 10%, have very much different halving times, going from a very rapid 11 days for the patient who went from 300% down to 1% to a relatively slow halving time for the patient who was already only at 10% when they started therapy. And that can happen for a patient at diagnosis. Some patients are at 10% BCR able. So their decline from 10% to 1 is only a 1 log reduction, so it's actually 27 days. So you need more than just the result at 3 months. You need the result at time 0 as well. But that can allow you to very accurately determine the halving time. And I think the halving time is actually stronger than the 3-month molecular response at predicting outcomes, including the success of treatment-free remission. So this is data that Narani Shanmuganathan put together and published a couple of years ago in Blood, showing that the patients who attempted treatment-free remission, now all of these patients were eligible, they'd all had at least 2 years of MR4.5, but the patients who had sustained treatment-free remission had a median halving time of 10 days compared to the patients who were unsuccessful where it was closer to 20 days. A very dramatic difference between the two with a very strong p-value. So in fact, if you look at the impact of the halving time on the success rate of treatment-free remission, if you look at the worst-case scenario, those patients shown at the bottom there who had a halving time greater than 21 days, even though they had had the same amount of time of therapy, that had deep response, that had 2 years of MR4.5, their success rate was less than what was around 10%. So in other words, something that happened to these patients in the first 3 months of therapy has impacted on their chances of success when you've stopped 6, 7, 8, 10 years later, suggesting that we're setting up for success in these patients right from the start and really bringing in what I believe should be our main focus to maximise treatment-free remission is achieving early, deep molecular response. If we can get short halving times, then we know that the success of treatment-free remission is much higher. And it doesn't seem to matter whether your halving time is achieved on imatinib or nilotinib or dasatinib. If you've got a long halving time, your outcome in terms of chances of success are equally poor. And likewise, if you have a short halving time, regardless of which drug you're on, your chances are equally good. So Narani recently has summarised all of the halving time data from all of the trials that we've run in Adelaide, right back to the IRIS trial, the TOPS trial, and beyond. So we have patients who received imatinib 400 milligrams, 600 milligrams, 800 milligrams, and then we have nilotinib and dasatinib. And if you accept the fact that the halving time is probably the best predictor of nearly all of the outcomes, you can see the very major impact just of increasing the dose from 400 to 600 brings that median halving time down from 18 days down to about 12 days. Interestingly, it's no better. In fact, it may be worse on 800 milligrams because patients frequently get dose interruptions because of toxicity on 800 milligrams. 600 milligrams seems to be the optimal dose of imatinib. I know that's not commonly practiced, but that's certainly the observation from our study. If you look at the more potent drugs, nilotinib and dasatinib, this is also based on the on the Australian Australian cohort of patients, you see, I'm not sure that's significantly different, but there seems to be a trend towards a lower halving time for the more potent drugs. And most importantly, I've shown there a cutoff at day 21, because we know patients with halving times longer than 21 days have a very poor chance not only of being eligible for treatment-free remission, but of achieving treatment-free remission. You can see with the more potent drugs that it's pretty unusual to have halving times longer than 21 days. So this is why I think we're going to see much better outcomes as we follow up our patients receiving frontline second-generation drugs. Now, there's a lot of interest and excitement about Asciminib, which you've probably heard about. Most of you are working with this drug. And its unique characteristics are that it's an allosteric inhibitor. It inhibits BCR-ABL by binding to a specific site, which is distinct from the ATP binding site, which has many potential advantages. Probably most importantly, the toxicity profile is very, very good. And I'll be presenting the data from the frontline study from Australia and New Zealand tomorrow morning in the oral session. But just very briefly to show the data that was in the abstract, this is you'll be aware of the very positive data we have for the Asciminib in the third line, both from the phase 1 study, but more importantly from the Assemble study that Delphine Ray led, showing the benefit of using Asciminib compared to bosutinib. We've taken that promising data and now designed a frontline study where Asciminib is used as monotherapy, as frontline therapy, and patients remain on 40 milligrams twice daily as long as they maintain an optimal response. If they lose optimal response any time, the dose can be doubled. And if they have ELN failure, that is greater than 10% at 3 months or 6 months, or greater than 1% at 12 or 18 months, you can actually add a conventional TKI such as imatinib, dasatinib or nilotinib to the Asciminib and then continue therapy. That's the design of the study, and so far we've been able to analyse one of the primary endpoints is the early molecular response. So we have that data from 79 patients and as I said, I'll be showing this data in some detail tomorrow. But it looks very promising. This is the molecular response. You'll see that the early molecular response rate is over 90%. Remember that with imatinib 400, the EMR rate is about 70%. You see the rates of MR2 very high. Even major molecular response at 3 months is already almost 50%. And we're also already seeing some patients with MR4. So this looks very, very promising, but of course it's early days. You see the median PCR value over the first 12 months as well on the right-hand side. Now, while Asciminib looks very exciting, I think we need to be aware of the fact that dasatinib is also a highly effective drug frontline, and this recent comparison of dasatinib 50 milligrams to 100 milligrams intriguingly suggests that 50 milligrams, even though it's a lower dose, may in fact give you slightly better outcomes, or at least as good outcomes as 100 milligrams, certainly less toxicity and arguably deep response at earlier time points. Most likely because you don't interrupt therapy for toxicity, you give continuous therapy, and I think that really one of the big questions in the future is going to be comparing dasatinib 50 milligrams a day, which is very well tolerated and clearly very effective, to Asciminib as a frontline therapy, and I'm hoping that could be a study that could proceed at some stage. The Ask for First study will be comparing Asciminib to investigator choice, which will include dasatinib, but that will be at 100 milligrams a day. So I mentioned the halving times and the fact that 600 of imatinib does significantly improve the halving time, and probably get further improvement using a more potent drug like nilotinib or dasatinib. And I can just give you a brief look at the preliminary data from Asciminib based on the Ascend study. And once again, we see very few patients who have halving times above 21 days, which I think is really good news. It's at least as good as dasatinib and nilotinib in terms of halving time. Warning, this is just preliminary data. We need further analysis to confirm this, but it looks very promising. So I think that we all understand that to achieve best response in our patients. We need to optimise our reduction of leukemia using tyrosine kinase inhibitor-mediated leukemia depletion. But I think increasingly we also need to focus on modulating the immune system to assist that process. And we need to understand a lot more about that before we know how to therapeutically modify the immune system. I hope I've shown you that rapid halving times is an incredibly important achievement for a patient, setting them up for very good outcomes all the way through for most of those patients. I think we need to be looking at pharmacologically modulating the immune response, particularly in patients who don't have a favourable response. Maybe not necessarily just at the time that we stop, maybe at much earlier time points is going to have more impact. That's something to discuss. And of course, in some patients where we identify persistence at a time when we'd like to stop therapy, looking at ways we can further deplete the critical leukemic cells. So my personal opinion would be that one of our big challenges over the next 5 to 10 years is to make treatment-free remission the rule for our patients rather than the exception, to make it low risk so that it's very, very likely that a patient who stops will be able to maintain their response and not have to go through the difficult phase of positivity and going back on drug. And I think that we need to be achieving this within a brief period, ideally 3 to 4 years, maybe even earlier if we could achieve it, rather than saying that we want to merely wait 14, 15 years before we can get to that time point. So this is my personal opinion, and this is something we can discuss at the end. My hypothesis would be that a focus on universal rapid leukemia depletion and early immune modification will move us closer to curing CML over the next decade. And I think that's something that, that certainly our laboratory is focused on, and hopefully studies that look at this question will proceed over the next 5 to 10 years. I want to acknowledge particularly Narani Shanmuganathan, whose name seems to be crossed out there, who did all of the halving time work and has been responsible for a lot of the molecular work that's come from Sue Bramford as well, and together they've done a lot of this very important work, and also all of the collaborators in Australia and New Zealand. I want to also remind you that we have 2 iCMLf global programs. One of them is in treatment-free remission, and at this stage we have 41 collaborators that are joining us in this treatment-free remission alliance from 21 countries, and we're very keen to have others join us as well for this collaboration, trying to further understand treatment-free remission. In 2020, we committed $100,000 to developing a global treatment-free remission registry. We had a competitive application that was awarded to Torsten Dahlen and the Swedish CML team, who are now developing that registry. And they're now doing that in collaboration with SAHMRI+, which has been a great development. And I won't go through all the details, but it's been a successful collaboration with SAHMRI+. Many centres are now getting their ethics approvals and are starting to put together the data from their many patients with treatment-free remission. We're aiming to get a cohort of around about 3,000 patients where we have all of the critical data, and we can try and understand what are the key predictors of success and where we need to do further research to understand treatment-free remission in more detail. There's some challenge in terms of getting US sites involved in the Harmony collaboration, which we're still looking at, but we'll still work on that because hopefully there will be solutions or an alternative approach we can use for those sites. Now, we did a pre-meeting survey asking this question about what people felt were the challenges for the next 5 to 10 years for the iCMLf, and we had 24 respondents from a variety of countries, including mainly clinicians, and quite appropriately we got many, many comments about our achieving global health equality, and all of those things listed there are things that I've mentioned that we need to be focusing on, and we really can't say we're making progress if we're still not giving drug to patients across the world, and we're not having PCR available across the world, so it becomes a major challenge for all of us. But also there was a lot of interest in genomic analysis, allowing us to predict exactly who would do well on a specific therapy and who needed a more potent therapy. This is an area of great interest as well for the Genomic Alliance that Andreas Hockhaus and Sue Bramford are leading. The idea of actually identifying high-risk patients who might have high risk of resistance. Management of side effects was certainly raised and is very important. Can we actually predict patients who get more likely to get certain side effects from their genomic profile? The idea of combining therapy is certainly of great interest and needs further investigation. Blast crisis is an area that we don't have a lot of clinical trials running because it's very challenging, it's rare, but we clearly need better designed and multi-country clinical trials to answer questions about blast crisis. And treatment-free remission, a lot of interest in how we improve outcomes in treatment-free remission. New therapeutic approaches, clinical trials. How can we improve the diversity of clinical trials? I think that's a good question that we can discuss. Elimination of quiescent leukemic stem cells, something which may be dealt with by Mike. And the idea that we can actually cure CML if we can identify the critical targets for elimination. So there's some initial thoughts. And we'll now run through the next three speakers, Mike Deininger, Ted Braun and Jorge Cortez. And following Jorge's talk, after each talk we'll have time for some specific questions for that speaker, and then at the end we'll have a more general panel discussion. So we have 5 minutes or so for any questions you have related to the talk that I just gave. Charlie.
We all recognized when 100% was arbitrarily chosen as the baseline for every patient, that didn't necessarily make sense biologically, because you mix in people with a lot of leukemia, a little bit of leukemia, and everything in between. And I remember when I argued that, John Goldman told me to stop and sit down. But I think it's true. And the halving time is really interesting, but what you didn't show us, however, the people who started at 300% compared to those who started, let's make it 50% or whatever, the spectrum of CML, and does that halving time hold up along the spectrum?
Yeah, it's a really good question, Charlie. So there was a study from, I think, from the German group suggesting that the level at the start did have some predictive value. Narani looked at this in some detail and found there was no predictive value. Am I misquoting you, Narani? Where are you? Is that correct? Thank you. So yeah, we looked at that very question. It didn't seem to be any impact having a level of 300% versus 30%. All that really mattered was that slope. Presumably the patient with 30% still has almost 100% leukemic cells. In fact, I'm not sure we've proven that, but I think that's the case. It's just that there's presumably less expression from each cell, because that, when we do DNA PCR, we find all those patients have very similar amounts. So that some patients presumably have less expression, it doesn't seem to have a prognostic importance. All that matters, and that's now, I think, convincing. I'd love to see someone. We did validate that data with the South Korean group, but we'd love to see other cohorts look at their data and try and validate it. It's tricky if you have ABL as your control gene, because you don't get that same sort of capacity to look at the starting level. But you can do it with any other gene, and we're looking at the moment to see whether using the Cepheid system, which is also based on ABL, whether that can give you a halving time which is maybe not quite exactly equivalent, but could still be predictive. So that's still work in progress. And Andreas would like to disagree with me.
Microphone is with FX. Hello, this is Jorge.
Oh, sorry, Jorge. I didn't see you over there, Jorge.
Thank you.
I'm not disrespecting you.
No, Tim, what you just said is really important. It's the control gene. We discussed this a lot in the past, and I'm not sure if Cepheid can overcome this. Unfortunately, the only FDA-approved test is the molecular MD kit, and this kit contains ABL as well. That means in all the studies which companies do, for instance, the Asciminib trials, all the Novartis-based Asciminib trials use the molecular MD system, and they will not be able, and that's important because they expect, Novartis people expect to be able to calculate the halving time, which is incorrect. Because the starting point is in most patients between 50 and 100%, and not the 300 you just mentioned, which makes biologically sense. And therefore, I think we should find a way to use alternative control genes for this specific question. And that's what we currently do in our investigator-initiated studies in Germany.
Yep, you're absolutely right. So, of course, it'll be possible, the RNA is there, it could be reanalyzed using one of the more appropriate control genes. Sue Branford at the moment is doing a parallel study, Andreas, where she's looking at calculating halving times and all of these patients on the Asciminib study using her in-house method, but also using the Cepheid and comparing. I suspect we're not going to get one-to-one, but you may still be able to identify those patients with you're not so sure, but I think we need to see the data. We need to see the data, and then we can argue again. Jorge.
Thank you, Tim. Very nice presentation. One question for clarification and another one more of a getting your thoughts. When you presented the data of the cumulative incidence of the deep molecular responses and sustained and all of that, I'm assuming that takes in consideration patients who change therapy and still yet make it there.
Absolutely.
And that's important because it's higher than what we've seen. I mean, the best data that we have so far from other clinical trials is the NSTND that goes to 10 years. But the sustained MR 4.5 is not even 50%. But that's on trial. It doesn't take in consideration that change in therapy. And of course, patients who have resistance are probably less likely to get there. But patients who change for intolerance and things like that will get there. So, so that's encouraging because it's more. Now, but here's the other question then. You showed how with the more intensive approaches, whether it's higher doses or with the second generation, hopefully the third generation, we're going to get a faster halving times. But there's still a big range. How much do you think we're going to make a difference? You know, my thinking is that there are some that we're going to be making a difference in the end. It's not just the halving time, but it's going to mean something in the end. But others are probably just going to go quickly, others probably won't even, but there's still the end result may not be the same. You know, is it biology, the halving time, or is it how hard you hit them? And I think it's probably a little bit of both, but I'd love to hear your thoughts.
Yeah, great comments, Jorge. Yeah, I mean, when I first saw it, I thought it was biology alone. When I first saw it, Jorge, I thought it was biology alone. So then we asked the question, well, but with nilotinib and Asciminib, you're getting these patients with dramatically better halving times. But is it equivalent to have a halving time of whatever, 20 days on imatinib and nilotinib or Asciminib? And correct me if I'm wrong, Narani, but it looks very similar. So that you actually can achieve a better halving time and also a better outcome. It's not just telling you something about the biology of the disease. It's a combination, presumably. And I presume that, I mean, I think the Asciminib data looks very exciting. I suspect, and it's based on not a lot of evidence, that I think we're making the good patients better. I'm not sure we're making the not so good patients that much better. And maybe those patients might need a combination up front. I don't know, but that's what we're sort of looking at the moment. Delphine.
Thank you. My question is maybe very stupid, but I take the risk of asking it anyway. Since we have this problem of the control gene at diagnosis, and most of us use ABL and are not ready to switch, have you looked at whether it's really between diagnosis and month 3? Or a little bit later, let's say between 3 months and 6 months, could we learn something about that, or is it already too late?
Yeah, we sort of asked that question. I'm not sure we've got too far with it. Do you have any comment, Narani? Have we ever looked at the 3 to 6 month? Can someone give Narani a microphone?
So we did look at between the 3 and 6 month data. The vast majority of patients, the slope certainly changed substantially by that point. So we didn't seem to see as much of a change and as much of a significant change after the 3 months. And we looked at even earlier time points as well, and we just couldn't really make much of it at that point. So certainly needs more looking at, but from what we've seen already with our cohort, haven't really been able to make much more of it.
Yeah, the other question I think I asked at one stage was whether you could, if you've got a problem with the starting, if you're taking a 2 and a 3 month, would that be enough to give you the slope? Did we look at that? I think we tried.
We tried, but it was a bit too untidy, too much of it.
So yeah, I mean, I think there's a lot of information in halving time, and I think it's frustrating that we don't have that data from big data sets for a kind of trivial reason. So I think we need to overcome that.
Yeah. You know, I was actually impressed at how small the range was in halving time.
How small the what?
The range was at 10 to 20 days.
Oh, no, there are a few outliers.
Yeah, no,
yeah,
but
you're
many. Clustered, and your means are one's 10 days, one's 20 days approximately, yeah. Right? And that's really very small. And, I'm thinking, you know, as I'm talking, but that seems to be a very small difference, and I'm trying to understand why. I mean, some of it could be, you really want to know the stem cells, I think, right? The proliferative compartment, the having time there. And it's possible that even though they have normal white counts by 3 months, some of the contribution of transcript is from more mature cells in the marrow. But without guessing, because it's the first time I've seen these data, I think that 10 to 20% is actually very small. And it's interesting that it looks like you're seeing differences with that small a difference in having time. And I would suggest people smarter than I think about that.
Well, I mean, we did a pretty basic thing, was basically we divided patients into quartiles according to their halving time. Was that based on the 400 or the 600 milligram data, Narani? 400. So we basically determined our quartiles from the 400 milligram data and then looked at the outcomes in patients in each of those quartiles, and I kind of stunned to see the outcome for patients who were greater than 21 days. I mean, it's terrible. There's almost no point stopping therapy in those patients unless we do something different. Bob's got a question. This is going to be a challenge, I'm sure.
I'm afraid I may be thrown out for spoiling the party. But I'm reminded of the Latin phrase, post hoc ergo propter hoc.
Of course you are.
So, I mean, these are they're both related statistical issues. But, and I've said the first part before, I mean, if you take people who've been in deep molecular remission for 2 years, 3 years, 6 years, 8 years, you will always find that TFR is increasing.
Yep.
And it's from selection bias. I mean, it's inevitable. It's not necessarily because they were in deep molecular remission for longer intervals. But the second, I mean, you don't need to respond to that because we've discussed
that discussion.
The other thing, the other point though, is that, you know, rapid response can be a selection factor, right? I mean, it identifies people who respond. It doesn't necessarily mean that it's because they're deep responders. It's identifying a population that's responsive. So, I mean, in order to sort this out, and maybe you've done it, in the slide you show with the different panels, if you take patients getting different drugs or different doses of drugs who have the same deep molecular, the same response,
yep.
Do they have different rates of TFR?
Yep.
Because otherwise it's just selection.
Yep. No, exactly right, Bob. And that's exactly the study that I asked Narani to do, and that's exactly what we found was that it didn't matter whether if you had a halving time of 25 days, it didn't matter what drug you had. You had the same prediction for TFR down the track. And likewise, if you had a very short halving time, it didn't matter whether you had that on imatinib or dasatinib or nilotinib, the same. But the only difference is you had a lot more patients with those short halving times with the more potent drugs. So it's always going to be a combination of things, I think. But the other question you raised, we need to have a formal debate on that because we keep coming back to it. I think I should stop there, and I'm going to invite...
Let me. Of course, I cannot say that so elegantly as Dr. Gale, but I wanted to ask the same question, but since it's been debated already, I'm not going to do that. So, but do you think the data assuming that this is disease modification, because this is what you're asking, is it disease modification or are you identifying a population? So if it's disease modification, this would argue that something happens over a 3-month period of time that doesn't happen or is less likely to happen with very potent TKIs and more likely to happen with less potent TKIs. So, and that makes a pretty big difference in TFR rates, right?
Yeah.
So conversely, if we leave a patient untreated for 3 months, I think most of us would think it's probably not going to make that big of a difference. Or even if you remember the IRIS trial, patients were allowed to have hydria for up to 6 months.
Yeah.
So do you think that you could do some kind of analysis to use that analogy to figure out whether or not this is disease modification or whether it's essentially just picking out the more favorable patients?
Yeah, well, I mean, most of our patients start pretty soon after they're diagnosed, but not all. Are you suggesting that that duration of from diagnosis to therapy might have some predictive? I think there was an analysis from IRIS that looked at that question, but since then we've been so rapid in our starting that we can't do that. I think in IRIS it did have an impact if patients who had longer than whatever it was did seem to have slightly worse outcome. But I don't think that's what we're seeing here. I don't think the fact that they have a long halving time is equivalent to having no therapy for 3 months. I'm not arguing that. I'm not saying, oh, that's allowing the clone to evolve over that period. I think the predominant reason for a short halving time is the biology, a favourable biology. But you can improve that by a more potent drug. That's what I think I would have to say.
But wouldn't be another argument to say that unless you hit it hard, it does something that you cannot correct at a later stage. And it doesn't do that if you don't treat it. Yeah. So in other words, you look at some reaction to the perturbation of the system.
I wonder whether, in fact, if you get that rapid reduction, it might then prime the immune system in a more effective way. And that's what we need to kind of correlate and work out. That's a hypothesis.